So plant hormone, or we can also say the plant growth regulator. Okay, so we know that the hormone are what? The hormones are the signaling molecule which is produced in a very low concentration or the low quantity in the organism's body. So whenever they are secreted, they trigger some responses in the target cells and the tissues by binding to the specific receptor. For example, in the case of animal, we know that the hormones are particularly they are transported through the circulatory system. Okay, but the plant biologists they argue that the traditional hormone concept it is limiting for the plant due to the difference in the physiology. So plants they do not have the circulatory system. Since they lack the circulatory system for the hormone transport, and some plant hormone they can act only locally, only means they cannot be transported from the one place to the another place. So they are working at the locally position or at particular point only. So that's why the term the plant growth regulator is preferred by many plant biologists to describe the compound that basically control the physiological functions of the plant. Okay, so despite the differences, the plant hormone can profoundly they can affect the growth and the development even in a very small amount. Okay, so they can affect the growth and development, and almost every aspect of the plant growth and the development is under the hormonal control. The hormones they have the multiple effect depending on the factors like the concentration and the developmental stage and the interaction between the different hormonal. They sometimes often control the growth and development rather than the isolated hormonal action. So let's see what are the different types of the plant hormone or the plant growth regulator. So these are the different types of the plant growth regulators: auxins, cytokinins, gibberellins, abscisic acid, ethylene, and besides that brasinosteroids are there, jasmonates are there, lactones are also there. So in this lecture, we'll study about the auxin, how it is get synthesized, how it is get degraded and transported to the various particular plant structure. So auxin, basically auxin is the first plant hormone we will consider because it deserves the pride of the place in any of the discussions of the plant hormone, because it was the first growth hormone to be discovered in the plant, and much of the early physiological work on the mechanism of the plant cell expansion they were carried out in relation to the auxin action only. Okay, so let's see the discovery of this auxin hormone. So in this diagram, we can see that the different experiment was set up by the different scientists: Darwin in the 1880s, and the Boyson and the Jensen in the 1913, the Pal in 1990, and then finally the Wendt in 1926. And Wendt 1926, the credit for the discovery of the auxin goes to the Wendt only in 1926. So from the experiment of on the coleoptile phototropism, the Darwin in 1888, he conducted an experiment, and that the growth basically or the growth stimulus is produced in the coleoptile tip. Okay, and is transmitted to the growth zone. So here in this we can see that the curvature, the intact seedling, when the light basically act on that particular side, then intact seedling curvature is observed. Now when the top of the particular part of the plant is when the tip of the coleoptile is excised, no curvature is seen. Similarly, the opaque cap is placed on the tip, then also the no curvature were observed. So from the experiment on the coleoptile phototrophism, it was concluded that the growth stimulus it is produced only in the coleoptile tip only, and it is transmitted to the growth zone. The Charles Darwin and his son, basically the Francis, conducted this experiment, and they studied that the plant growth phenomena, including the tropism. And he observed that the bending of the plant toward the light using the canary grass. He performed the experiment on the canary grass. Okay, so we know that the coleoptile especially are very sensitive to the blue light, and they bend toward the light source when illuminated on the one side, and the tip of the coleoptile they perceive the light, but the growth zone responsible for the bending is several millimeter below the tip here. Okay, so he concluded that the signal which is produced in the tip, they travels to the growth zone using the differential growth. Next, in 1930, the Boyson and the Jason, they discovered that the growth stimulus, they passes through the gelatin but not through the water impermeable barriers such as mica. Because we know in this experiment, they performed that the mica sheet when inserted on the dark side, then the no curvature were observed. But when the mica sheet is inserted on the light side, the curvature were observed. Now, when the tip were removed, then also the no curvature was seen, and the gelatin between the tip and the coleoptile were placed. Then also the no curvature is this. Then the normal phototrophic curvature remains possible. Okay, after some time with this. So they concluded that the growth stimulus they passes through the gelatin, this gelatin. Okay, because when he placed the gelatin, it was assumed that the stimulus they must have passes through the gelatin, but not through the water impermeable barrier such as the mica. Okay. In 1990, the PAL provided the evidences that the promoting stimulus produced in the tip was chemical in the nature. Okay, so in the first experiment, the tips were removed, then the tips were placed on the one side of the coleoptile stem. Still, the growth curvature developed without the unilateral light stimulus. Okay, next, at last, the Fritz went 1926. He demonstrated the presence of the growth promoting chemicals in the oat, that is, oat plant or oat coleoptile tips, and the previous attempts were actually attempts to isolate this chemical. They failed due to the release of some inhibitory substances during the tissue grinding. So what actually he did, the coleoptile tips were grown on the gelatin, and then the tips were discarded, and the gelatin cuts up into the smaller block. When this gelatin block is placed on the one side of the coleoptile stem, then the coleoptile bends in the total darkness. Angle of the curvature can also be measured. So the vent breakthrough involved allowing the chemical to diffuse out of the excised coleoptile tips directly into the gelatin blocks, and when the gelatin blocks they were when placed asymmetrically, like on one side only on the decapitated coleoptile, they causes the bending indicated the presence of the growth promoting substances. Some growth promoting substances are there in their coleoptile tips. Okay. So these substances, they were eventually named auxin from the Greek word which is